The key question we want to address with this method is, how can we use RNA samples from material purchased at local food markets for the cloning of biotechnologically relevant genes? Here, we use specific oysters as a case study for demonstrating this approach and potential further application.
The main advantage of this technique is that conventional genome or cDNA sequencing of the oyster samples can be circumvented. Instead, sequences of the cytochrome C oxidase subunit 1 and NADH dehydrogenase were compared with publicly available reference sequences from the Pacific oyster to select the most closely related specimen. The cDNA prepared from this specimen can be directly used for the cloning of biotechnologically relevant genes, which can be selected using the public sequence information.
Demonstrating the procedure will be Yong and Yuchuan, two graduate students from the Glycomics and Glycan Bioengineering Research Center at Nanjing Agricultural University.
To prepare oyster tissue sample, use a sterilized scalpel to cut out approximately 100 milligrams of homogeneous soft tissue from the approximate geometric center of each oyster specimen. Transfer the sample into a mortar filled with 50 milliliters of liquid nitrogen. Grind the flash-frozen oyster tissue into a fine powder. Weigh out 75 milligrams of each specimen's frozen tissue into a sterile 1.5 milliliter centrifuge tube and mix with 1 milliliter of guanidinium thiocyanate phenol extraction reagent.
The most critical step of this procedure is the RNA